Hello, welcome back. We are now moving on to the central nervous system brain video. So in this video, we're going to focus on structures and functional regions of the brain, and then finish up taking a look at the things that are there to protect the brain, like your meninges and cerebrospinal fluid. All right. So let's just take a look at the brain. We're going to see this picture a lot. I'm just going to use it repeatedly, but then highlight the different parts of the brain that we're going to take a look at. So I think I'm going to use a highlighter for this because they kind of do a little bit better job of when I do big drawing type of things. So when we take a look at the brain, it has four major regions that I'm just going to highlight here first. So the first one, this highlighted in green, this is the cerebellum. Sorry, the cerebrum. I was thinking cerebrum, but said cerebellum. So that's the cerebrum. It's the biggest part of the brain. Okay, so that's your cerebrum. The next part is what's called the diencephalon, and that's this middle part here. We'll go back to pen for writing. So that's your diencephalon. It's kind of the part right at the very top of the brain stem, which is what we're going to take a look at next. So brain stem is this part right here, kind of highlighted in yellow, all that kind of greenish part there. So this is your brain stem. And then the last region is called the cerebellum. That's the cerebellum. So we have the cerebrum on the top. We have the diencephalon kind of deep in the middle. We have the brain stem is kind of like the stem, like the stalk. And then the cerebellum actually means little brain, little cerebrum. So the cerebellum is back here, kind of where your occipital bone is. So those are the four regions of the brain. So we are going to be taking a look at these as we go through the different regions. So we're going to hit the cerebrum first and take a look at some of its features. Okay. So that's kind of this color-coded cerebral hemispheres here. So your cerebrum is broken up into two hemispheres. You have your left and right hemisphere. There's a nice longitudinal fissure that kind of separates the two. And then there are lobes, cerebral lobes. And if you'll notice, their names look like your bones. So the frontal lobe sits under your frontal bone. Your parietal lobe sits under your parietal bones. Your temporal lobe sits under your temporal bones, and your occipital lobe sits under your occipital bone. So that's really nice because that's bringing back your prior knowledge of your bone list. So that helps to learn these and makes it a little bit easier. So those are the major regions of the cerebrum. They're broken up into those different lobes. And then just some vocabulary that goes along with brain anatomy and especially cerebral anatomy is, if you'll notice, the cerebral hemispheres are very wrinkly. They have all these kind of wrinkles built in. The wrinkles actually have names, of course. They are called a gyrus, is like the ridgy part, like the bump part, and the sulcus is the dips, right? So a gyrus, sulcus, gyrus. So all of the little ridges are called gyri for plural, and sulcus for singular, or sulci for plural. And then sometimes you might have a fissure where the fold goes really deep, and a lot of times all right, I had a little bit of technical glitch there, so hopefully everything's going to go smoothly. So this next slide is taking a look at the functional anatomy of the cerebrum. So we just labeled all of the different parts, so our different lobes. So what's happening in those different lobes? Well, we're going to take a look at the frontal lobe first. So this very frontal part, kind of this hot pink area, kind of like right in here, ignoring the Broca's motor area for a second. So this very front part of your brain is for language comprehension, critical thinking, consequences, risky behaviors, predicting what's going to happen based on your behavior and your choices. And so this is kind of the mature adult brain, if you will, because it's the last part of your brain to fully develop. So your brain kind of develops from kind of posterior and inferior kind of this way. And so children and young teenagers and older teenagers and young adults sometimes seem to kind of participate and engage in risky behavior from an adult perspective because they may not have their brains fully developed. Now, that's not giving people a pass because they have other parts of their brain to know the difference between right and wrong, but the difference between fully comprehending the consequences of their actions is a little bit more challenging for underdeveloped brains. Another region that's associated with the frontal lobe is that motor speech area that kind of sits right here, a little bit anterior to your temporal lobe. So this is the area that is a connection between all the neural control of the muscles of your tongue and your mouth and your diaphragm to allow you to speak. So if people have damage to that part of their brain, a lot of times that damage can happen in a stroke. They lose the ability to speak. They can understand, they can hear, they can want to say words, but then the words just don't form because they have damage to those neurons that control the muscles that allow you to speak. So unless they get physical therapy or kind of relearning and bringing in other neurons to take over that function, sometimes people never regain the ability to talk. We have the frontal association area right here, which kind of helps to connect some of those kind of higher thinking level stuff with your motor commands, because all of this red area here, kind of this area here, this is what's called your motor area and your premotor area. This is the initiation of all of your skeletal muscle action. It's like this band that sits right on the middle part, kind of middle forward part of your cerebrum. So anytime that you move a skeletal muscle, that signal initiates in the premotor area, gets traveled to the primary motor area, then sent down towards your spinal cord to send messages out to your limbs. Okay. On the parietal, occipital, and temporal lobes, these are dealing a lot with sensory information coming in. So we have what's called the somatic sensory area. So remember, somatic means conscious. So that somatic sensory area is bringing in information that we are consciously aware of. We have our gustatory area, kind of right down here on our temporal lobe. Gustatory is taste. We have our visual area back here, way at the back of our occipital lobe. And then we have auditory, so that's hearing, also kind of located on the temporal lobe. And we have olfaction, which is also located on the temporal lobe. So the temporal lobe is really busy for taste, hearing, and smell. The vision happens primarily back here in the back of your brain. We also have these other areas that are like general interpretation. You're processing a lot of information, making connections between all of these other different areas. And then we have speech and language, which is processing. The motor Broca's area, this motor speech, allows you to use the muscles to make words. These speech language areas allow you to comprehend words. So if there's damage there, then a person might have a hard time comprehending or being able to figure out which words to try to say. So the cerebrum is a very busy, complex place. So as you're learning these things, just learn them as their general description. So we don't have to go into too much detail, so the frontal cortex is for higher thinking processes, premotor areas for skeletal muscle, somatic sensory is for general sensations. Then we have hearing, taste and smell in the temporal lobe, vision in the occipital lobe, kind of that level of learning about these cerebral centers. Okay. So here's a cool picture to kind of show that primary motor and primary somatic sensory association areas, those two gyri that run kind of almost like headbands across the top between the frontal lobe and the parietal lobe. So for the primary motor area, what they've done in experimental situations or where a person is having brain surgery and their brain is open anyways and they consent to helping neuroscientists learn more about things. They can stimulate various parts of these primary motor and primary sensory cortices and basically map out what they control. So what they've done on this diagram is they've mapped out the different body parts that correspond to those different brain regions. So if you're a researcher and you are, stimulating this area right here, the patient's toes may wiggle. They're not telling them to wiggle, but the researcher is stimulating the brain. Or here, your fingers might wiggle, or they might blink if you stimulate here, or their tongue might be wiggling if you stimulate over there. That's the motor cortex. That's how they know, because they've actually poked people's brains and triggered those parts to move. On the sensory side, they would stimulate that post-central gyrus, that primary sensory area, and the person, because they're awake during these brain proddings, they would say, hey, if you're stimulating here, it feels like you're touching my fingers, or it feels like you're touching my lips, or it feels like you're tickling my armpits, or whatever the sensation is. So they physically map these out on people's brains, since that's how they know where these primary motor and primary sensory informations are found. So if there's damage to any parts of those brains, then they can predict that that patient might have some issue either controlling that part of the body or receiving sensory information from that part of the body. Okay. All right, so now let's move on. So that was our cerebrum. So now we're going to move on to the diencephalon. So let's highlight that here. So the diencephalon, I'm just going to talk about this part that I'm highlighting here. So ignore everything else. We're going to talk specifically about this. So the diencephalon is made up of three major regions. We have what's called the epithalamus. We have the thalamus. And we have the hypothalamus. And you might see that they have kind of a directional naming. So epi means above and hypo means below. So let's find the main thing, the thalamus. So the thalamus is, I'm going to just kind of, let's see, what would be a good color? Maybe the darker yellow. The thalamus is this blue kind of egg-shaped thing right in the middle. Okay. Think of the thalamus as a good example that I like to use is like the executive secretary of the brain. Okay, the executive secretary. So all of this information is going to be traveling up from all parts of your spinal cord, different parts of your brain, all of this stuff. And the conscious awareness, all of this stuff up here on your cerebral cortex, is really busy dealing with a lot of things. So if all of this information is coming up, the thalamus is kind of like the secretary that says, okay, you are important enough information, you get to go up to the president, the CEO of the body, which is your cerebrum, the cerebral cortex. But if not, I'll divert you over here, and this part of the brain will be able to take care of that. So I don't have to bother the big cheese up top to be able to handle that. So I'm going to divert that away. So it's like a relay station. If you think of those old telephone operators that have to plug in all of those different cords to connect people calling on the telephone. So that's what the thalamus does. It sends only the most important information up to the cerebrum and then reroutes all of the other information coming in to the different parts of the brain that can handle it. And so that's the job of your thalamus. The epithalamus primarily contains two major structures. It's going to contain the choroid plexus, which we'll see as part of a production of the cerebrospinal fluid, and the pineal gland, which secretes your melatonin, which is a hormone that helps you regulate your day-night cycles. So see this little kind of orangey red caterpillar looking thing? That's the choroid plexus. And then this little blob right here, that's the pineal gland. So those two things make up your epithalamus, sometimes considered the roof of your thalamus. Let me make sure I'm following along in the notes. Thalamus, okay. Epithalamus, choroid plexus, and the pineal gland. Thalamus is your relay station for this impulse. We talked about that. Okay. And so then the hypothalamus is this purpley structure down here. So the hypothalamus, and we'll include the pituitary gland with that. So all of this almost like funnel-shaped bird beak thing, that's your hypothalamus. It has a lot of functions. We'll see more, or you will see more of those, because I don't usually teach the 122. But when you get into 122, you'll learn a lot about hormones. You'll be doing the endocrine system, one of the first things you do when you get back in the fall. And the hypothalamus and the pituitary gland are big parts of that. This right here is the pituitary gland. My computer's kind of glitching today a little bit. So this right here is the pituitary gland coming off of the hypothalamus. So what we are going to cover in the hypothalamus is it's a big control center for a lot of things. So it's going to control temperature. So your internal thermostat is located in your hypothalamus. It is an autonomic reflex station. Running out of room. So autonomic reflexes, things, remember we just learned about reflexes in the last video. So reflexes that are dealing with cardiac muscle, your respiratory rates, your heart rate, your background level of like salivation or blood vasoconstriction, vasodilation, bronchoconstriction, bronchodilation, regulating a lot of your autonomic functions happens in your hypothalamus. Let's see, regulates body temperature, water balance, metabolism, also the center for your emotions, your thirst, appetite, pain, pleasure. Those things are all found in the hypothalamus. And then we have our pituitary gland. And then this little blob right here is called the mammillary body. It regulates reflexes that have to do with eating and chewing and kind of swallowing, all that kind of stuff. So it's a really busy place. So there might be a list in your textbook. I don't have it with me at home, but you can just briefly list, maybe bullet point list some of the things that are found in the hypothalamus. So temperature regulation, autonomic reflex center, motivational drives, emotional drives, your pituitary gland, and your mammillary body. So those are some of the key things that are found in your hypothalamus. All right, so all three of those things collectively together is what we call the diencephalon. Okay, so then the next thing we're going to take a look at is the stalk, if you will, because sometimes people think the brain looks like a big mushroom, and I guess it kind of looks like a big mushroom. So now we're going to take a look at this stalk. So this is our brain stem. So kind of moving from the top down, we covered our cerebrum, the diencephalon, and now we're going to take a look at the brainstem. So the brainstem has three major regions. So we have the pons, we have the medulla, oblongata, and we have this area here is called the midbrain. Sometimes called the mesencephalon. Your book calls it the midbrain, so I'll call it the midbrain. All right, so the midbrain has a couple different functions. So now that I labeled those, I'm going to clean up the picture a little bit. So midbrain, pons, and medulla oblongata. You can follow along in your book. So I'm going to then highlight the parts of the midbrain. So we have this area right here are called cerebral peduncles. It's a fun word to say. Cerebral peduncles. This is, think of like a big extension cord or a big phone line. It's just basically a whole bunch of ascending and descending neurons. Information going up to the thalamus, information coming down from the thalamus or the cerebrum. So it's just kind of a whole bunch of white matter, which is myelinated axons, a lot of stuff going up and down. Then we have this collection, these two bumps. It looks like two bumps from this perspective, but if you're to look at the full backside, there's four. So this is what we call the corpora, which means body. I'm going to run out of room, maybe. Gemina. Okay, corpora quadrigemina, which means a body of four bodies. So we have the two bumps. So it's going to, from the back side, if we're to look at it, it's going to have like these superior bumps and then these inferior bumps. So it's like 4 structures like this. And we're only looking at the 2 when we slice it in this lateral section. So these are called the superior bumps. They're not called superior bumps. They're called superior colliculus, but I don't think you go into that much detail. So this top set does vision reflexes, and this bottom set does auditory reflexes. So these are your vision and auditory reflexes. So think about, like, if you ever flinch, like something's coming at you, but you don't know what it is, and you kind of flinch a little bit, that is being regulated by these top set of bumps. Or if you hear a loud sound behind you and you kind of turn quickly towards the sound, that is your auditory reflex, regulated by the lower half of your corpora quadrigemina. Okay, so that's part of your midbrain. So cerebral peduncles and corpora quadrigemina. Okay, so that's the midbrain. The next part is, we're going through the list. Okay, so this is the pons. So the pons is this little bump that kind of sits right out front, a really good landmark as you're practicing labeling your brain and learning the parts and pieces of your brain. There's lots of functions of the pons, but we're not going to go into very many of them. We're just going to actually list one major function. There are centers in the pons, and they regulate breathing. Okay, so reflective centers in breathing. We talked about some of those autonomic functions in the hypothalamus. So there's actually centers up here that can come down and impact those regulating centers in the pons. And we're actually going to see there's more centers down here in the medulla, which then finally go down and tell your diaphragm kind of the pattern and the rate and the depth at which to inhale and exhale. So for now, be able to identify what the pons is, and then we're just going to focus on the function of breathing for the pons. And then lastly is our medulla, right? So the medulla is this kind of the bumpy part right below the pons. So our medulla is continuous with the spinal cord. So those axons, ascending and descending neurons that are sending this information from the spinal cord up to the brain, they travel through the medulla. And so in the medulla, we have, again, fiber tracts. So that's your what we call the ascending and descending tracts. We also have what's called the reticular formation. Reticular formation. Yeah. So it is a collection of neurons that kind of runs along the backside like this. Think of reticular formation as your circadian rhythms, your day-night cycles. It helps you to maintain consciousness during the day and helps you to go to sleep at night. We also see a lot more reflexes in the medulla. So more breathing, a respiratory rate, like I mentioned, heart rate, those very basic bodily functions are kind of maintained at these base levels in the medulla. It's what's called a primitive brain sometimes. So if people get damage up here in the cerebral cortex and their conscious thought is not there, say like they're in a coma, but if all of these lower level functions are still intact, then they can be breathing, having a heart rate, doing all that kind of stuff, but they're considered brain dead. Not that their entire brain is dead, just the conscious part. They can't pull up any of the functionality of like their frontal lobe or their occipital lobe, but their base functions are still working because your brainstem and diencephalon are intact. Okay, so that's your brainstem. So midbrain, pons, and medulla. Okay, the last part is of the brain is your cerebellum. Okay, this guy here, to me it always looks like cauliflower. It's like some branches of cauliflower. So the cerebellum functions primarily to maintain our balance in three-dimensional space. So it takes in a lot of information, processes that, and allows you to maintain postural muscle contraction, move your muscles, kind of keep you in balance with where you are at in three-dimensional space. So sometimes to help you understand what a part of a brain does is to talk about what happens when it's not functioning properly. And so imagine, sometimes we see this in movies or in news or maybe in real life, somebody who is very intoxicated and they can't walk very well. They kind of stagger and they're kind of walking, they can't walk a straight line, or like you might see in a sobriety test, because alcohol comes in and affects the neurons in your cerebellum, so they cannot do their job properly. So when somebody has damage or inhibition of their cerebellum, we can tell that their balance and kind of ability to recognize where they are in three-dimensional space has been diminished. So that's the job of the cerebellum, right? Okie doke. So those are all the parts of the brain. We still have a couple more slides left in this video to talk about protection of the brain. So how do we, all of this huge mass, it's the biggest collection of neurons that we have in our body, how do we protect that and keep them from being damaged? So the first structural thing that we do is have what we call meninges. Meninges are layers of connective tissue under your skull, but not part of your brain. These layers that help to protect your brain. There's three major layers. We have what's called the dura mater, which in itself is made up of two layers. We have the arachnoid mater, and we have the pia mater. Okay? So the pia mater is that kind of turquoise light blue. It covers and follows every curve, gyrus and sulcus of the cerebrum, and all the other brain parts as well. So that's going to follow every little curvature of the surface of the brain. The arachnoid mater is a light purple, and if you'll notice, there's a space there, right here, this subarachnoid space. This is filled with CSF, cerebrospinal fluid. So the cerebrospinal fluid helps to collect debris. It gets recycled on a daily basis, and it helps to cushion and protect the brain. So, you know, as our head is wobbling around inside of our skull, we have this little watery airbag, a water bag, this watery protection to keep our brain tissue from ramming into our surface of our skull inside. So the dura mater, the arachnoid mater, the pia mater all help to wrap the brain, but it's this thin layer of cerebrospinal fluid that helps to cushion and support the brain. That's why concussions are such a big deal, because your head is hit so hard that your brain actually can physically ram the inside of your skull. So that CSF-filled space is overwhelmed by the amount of trauma, and that physical impact of your brain touching your skull or hitting your skull can damage your neurons, and you might get a little, you know, bruising or bleeding or death to those neurons, and then you've lost that function because neurons do not repair themselves. Okay. So in this diagram, I'm also going to point out some things that we'll see on the next slide, next few slides, when we take a look at the production of CSF, because we have CSF, like I've mentioned, is filling in this space, this subarachnoid space, and I mentioned it's being recycled. So this box right here that I'm drawing, right around that kind of blue triangle, there are kind of large veins called dural sinuses, and this one's particularly called the superior sagittal sinus because it's running along the sagittal plane here. So there's little extensions of this arachnoid mater, these little purple guys I'm outlining right here. So they allow the CSF to kind of filter out of those little pockets into this venous blood. So we're continually making new cerebrospinal fluid at the choroid plexus, and it is recycled in these little pockets into these dural sinuses, like the superior sagittal sinus. Okay. All right, so then here we have these hollow spaces that are filling the brain. So remember our ependymal cells we saw way back in our neurons video that line these hollow spaces within the brain? Well, here they are. There's four major ventricles. We have two lateral ventricles. Okay, so we see them in these two perspectives. We have a single third ventricle, kind of centrally located, which drains down into the fourth ventricle, which is also centrally located. And they are connected by what's called the cerebral aqueduct. Okay, this little tube inside. So these ventricles, so we have the fluid that's filling that subarachnoid space around to kind of help give buoyancy. And then we also have this fluid deep inside of our brains that also helps to give protection and buoyancy to the brain that's kind of floating in your skull case. So looking at how and where this CSF is produced, we have, remember we just identified the choroid plexus in the epithalamus. We also have a little bit of choroid plexus down here in the fourth ventricle. So as the choroid plexus is producing the CSF, you can see those little black arrows. It flows into that lateral ventricle, into the third ventricle, down the cerebral aqueduct, into the fourth ventricle, down all the central canal of the spinal cord, which will be another video. It leaks out into that subarachnoid space, right? So that's this kind of light blue area here. And then remember those little projections that I told you? These are called arachnoid villi. So that is the recycling into that superior sagittal sinus. So we have the production by the choroid plexus, circulating through all of the ventricles, circulating through this subarachnoid space, and eventually drains into the dural sinuses. Okay, so that's the flow and kind of pathway of the production, flow, and recycling of your cerebrospinal fluid. So make sure you are comfortable. Maybe just label a picture like this or draw or outline the steps of CSF production, flow, and recycling. Okay. The last couple of slides are, this one is a little bit more clinical. You may have interacted with this process before. So there's a couple different things that are going on here. So if we wanted to take a sample of that CSF, we can do what's called a lumbar puncture, or sometimes called a spinal tap, in which a needle, a big long needle, goes in between your vertebrae, penetrates through the dura and arachnoid mater, and retrieves CSF fluid from that subarachnoid space. And they usually do this test to look for infection. So this is what we call a lumbar puncture. It has to be done by a very highly trained anesthesiologist or a doctor to be able to pull out that fluid without damaging your spinal nerves. The other thing that somebody might have been exposed to is in the spinal cord, there is a little bit of space called the epidural space. It's outside of the dura mater, right here in the space here. We can kind of see it out here as well. So it's just outside the dura mater. So this is the target of anesthesia during an epidural. So if you've ever heard that, or a spinal block. So this is an epidural. So a lot of times this process is done for women to help reduce the pain during labor and delivery. So that is associated with this, the dura mater, the meninges, and this particular region of the protection of the central nervous system. And then the last thing we're going to talk about is the blood-brain barrier. So neurons are very picky. They only metabolize glucose. They don't really break down proteins or fats or anything for their ATP. So they only rely on glucose. And they don't like a lot of other stuff just being able to go into the neurons willy-nilly. So we have, if you remember, tight junctions way back from epithelium. So tight junctions are so tight that water even can't get through. So here's a nice, simple squamous epithelium, kind of bounded to itself with tight junctions right here. So the only way something can get from the inside of the capillary outside of the capillary is through facilitated diffusion. And if these cells don't have a channel, you're not getting out. So that's kind of what this image is showing you. That big gray molecule cannot leave the capillary. It just is going to bypass and go back into circulation, and it's not going to leave the blood and go into the brain. We also have all of these astrocytes that completely cover these capillaries, and they themselves are connected with tight junctions, so nothing's going to leak out. So it's almost like a double layer of protection. We have the endothelial cell from the capillary, we have the projection off of the astrocyte cells, and so if you are something that needs to leave the blood, you have to go through the epithelium and you have to go through the astrocytes. So it's very well protected and very well guarded. So only the very specific substances, nutrients, and things can get through that blood-brain barrier. All right, so that wraps up our brain section. So we are going to move on to the spinal cord. Bye, I'll see you next time.